Thank you for giving me this opportunity. A part of my presentation today is the
ano to yung
result
mic ko?
obtained by Professor Fema Abamo, Mindanao State University. She was a graduate school student of my laboratory at Yamaguchi University, Japan. The serious parameter plays an important role in maintaining the quality of fresh water and brackish water by ingesting small particles including
ano to yung
bacteria,
camera ko?
small algae,
mic
less
ba?
than 10 micrometer
No, layo lang.
length.
Microphone.
However, recent rising water temperatures due to the global warming are predicted to inhibit paramecium survival and causing a decline of water quality in ponds and lakes. Therefore, if we can make paramecium high temperature resistance in the Therefore, if we can make paramecium high temperature resistance in a safe and maintenance-free manner, we can expect to use their function for water quality conservation even in the elevated water temperatures. At first, I would like to introduce Yamaguchi University Resource Center for some tolerant microbial resources, where I am now belonging as a visiting researcher. This center consists of 3 divisions, and I am now a member of the Division of Environmental Microbe. So if you have an interest or collaboration with this research center, please contact here. Yamaguchi University is here, about 5 hours from Manila. Not so far away. This is the paramecium cordatum. Cell length is about 200 micrometer, divided 3 times per day at 25 degrees, and living in fresh water. Paramecium cordatum has a small germinal micronucleus and a large macronucleus and big oral apparatus and many digestive vacuoles and a side blocked and 2 contractile vacuoles and many trichocysts and cilia like this. Paramecium uptake bacteria, algae, and small particles from this oral apparatus into the digestive vacuole and discharge undigested materials from the side product. Therefore, paramecium is an important member for water quality preservation in pond lakes and also in sewage treatment plants. For example, left side is tube culture in log phase of growth of paramecium cordatum. The culture medium is cloudy with food bacteria. Right side is of stationary phase of growth. After 24 hours at 25 degrees, food bacteria are almost digested, and as you can see, culture medium became clear. We can see many paramecium cordatum as a white dot in the test tube. In this way, paramecium contributes to water quality preservation by its strong phagocytic activity. However, paramecium cells decrease cell division speed at 30 degrees and dies at 40 degrees. Therefore, rising water temperatures due to global warming will hinder the survival of paramecium cells and induce a decline in water quality. However, fortunately, we found paramecium cells infected with end-nuclear symbiotic bacteria Holospora are transferred to high-temperature resistance. I will explain at first how Holospora transforms its host paramecium to high-temperature resistance, and then the possible use of Holospora-infected paramecium for keeping the water quality in global warming conditions. At first, I have to explain about Holospora. Holospora species is a gram-negative end-nuclear symbiotic bacteria of the seriated paramecium species. Holospora shows two different morphology in their life cycle. Before that, I forgot to explain about this figure. This is Paramecium corditum. As you can see, the many bacteria. This is Holospora obtusa, macronucleus Paramecium corditum specific bacteria. Here, as you can see, many bacteria. And this bacteria shows two different morphology in its life cycle. One is infectious long form. The length of bacteria is about 30 micrometer in length, very long as a bacteria. And this is a reproduction. During infection process, infectious form always escape from the host digestive vacuole from the with this invasion chip, and also penetrates their target nuclear envelope with this invasion chip. So we named this chip as an invasion chip. We can isolate the holospora from the host homogenate by Parcour density gradient centrifugation. This is isolated infectious form of holospora obtusa, bright region under phase contrast microscope is a cytoplasmic region with two DAPI positive nucleoid, and the dark region is a periplasmic region. This is isolated reproductive short form and the intermediate form in differentiation to the infectious form. They have no invasion chip, and also DNA is distributed in 4 cells. This slide shows the Paramecium corditum becomes high temperature resistant by infection of Holospora obtusa. This is 2-second swimming traces. When Holospora free cells and Holospora bearing cells were which were cultivated at 25 degrees were transferred to 40 degrees. As you can see, Holospora free cells stopped swimming and dies immediately. However, Holospora bearing cell here, they can swim, survive. Namely, the host cell acquires high temperature resistance by infection of this Holospora obtusa. Then, how can Holospora change the host gene expression? We found infection of Holospora obtusa induce overexpressions of the host heat shock protein genes, HSP 70 and HSP 60. Compared to the Holospora free cells, you can see many mRNA appeared. This suggests overexpression of these two genes is responsible for the host acquisition of the high temperature resistance by infection of Holospora. So we examined how can Holospora induce overexpression of these host genes. We hypothesized some substances secreted from Holospora may affect the host gene expression.
Okay, allow me to read the content of the plaque. Mindanao State University Maguindanao present this award of appreciation to Professor Dr. Errol Acoso, who's represented by Professor Dr. Gunchuk. Yeah. Given to Professor Dr. Masahiro Fujisima. Yeah, all right. Maraming salamat po.